Good afternoon, everyone. My name is Stéphanie Chevalier. I am a PhD student in the University of Paris-Saclay, and I represent this work with Vincent Noel, who is a postdoc fellow in Institut Curie in Paris. We present a methodology for synthesis and simulation of ensembles of Boolean networks for self-aid decision. The context is that computational models of molecular interaction networks are usually built from data on the structure of the biological system, so structure of the interactions that are implied in the process we want to model, and also on data on the behavior of the biological system, so its dynamics. The structure of the biological system constitutes the static part of the data for the modeling. It's the known and putative interactions between biological entities, as you can see here. Biological entities that can be genes or proteins, for example. This information constitutes interaction graphs, which are prior knowledge networks that delimit the domain of interactions that can be included in the models. On the other hand, the behavior of the system is described by observations on the system and the dynamics of these observations. It can be measurements of gene expressions or proteins activity at different time and or under various conditions. Thanks to these dynamical observations, constraints on the dynamics of the models arise, because model dynamics have to contain the same sequence of observations, the same observed stabilities, for example, and so on. But the synthesis process encounters an important issue. The observations of the biological processes stay very scarce, either in terms of temporal resolution, number of observed entities, or variety of experimental conditions. And this problem is combined with complex and large structures for molecular interactions. So the model engineering problem is often largely underspecified, and this leads to many potential candidate models, often too many to be enumerated exhaustively. It's why we present an ensemble-based approach to Boolean modeling, because by constructing and reasoning on dynamics of ensembles of Boolean networks, we aim at reducing biases in subsequent analysis, subsequent predictions. Our strategy is at first to synthesize Boolean model ensembles. Ensembles representative of the multitude of models compatible with both network architecture and dynamical properties. And secondly, we want reasoning on the dynamics of the ensembles of models by running simulations taking into account all these models. To do so, simulations are aggregated through averaging. It's particularly interesting because it allows an interpretation at the level of cell population and takes into account its potential heterogeneity. We can then quantify the propensities of reachable attractors possibly subject to network perturbation. We will present at first our synthesis method based on the logic framework of Ensemble set programming, and then our simulation method based on ensemble of models. And we have illustrated this methodology on a model of molecular pathways regulating tumor invasion and migration. We evaluated the shift of reachable phenotypes caused by interaction between mutations. Let's start with some definitions. A Boolean network is a function which takes and returns a vector of Boolean values, vector called a configuration. The Boolean network is constituted by a set of low-code functions, which are logical rules, one per component of the Boolean network. They take a configuration and return a Boolean value. Therefore, the activity of components is simplified to off and on. For example, you can see here that the gene 1 can be active only if its inhibitor gene 2 is inactive. So a value can be computed for each component given a configuration, but to draw the dynamics of a Boolean network, a semantics is necessary. because semantics specify how to compute the next possible configurations. For example, updating only one component at a time, as in the asynchronous semantics. The one we consider for the synthesis is the most permissive semantics due to the guarantees it provides, and also the lower complexity for deciding main dynamical properties. This semantics is described in a Nature communication published in August. You have here a link toward the article. I will start describing our synthesis method. We formulate the problem of Boolean network synthesis as a Boolean satisfiability problem implemented in Ensemble programming, also called simpler ASP. In fact, we have extended our prior work on Boolean network synthesis. You can see here the reference of the paper, where the method and its implementation is described. It included several constraint types, such as existence of a fixed point, attractor properties, and irreversibility of amplification, all of this considering the most permissive semantics. It's really it's interesting to be able to model cell differentiation, for example, because these constraints match well the observed properties of cell populations evolving towards mutually exclusive phenotypes. This approach leverages both a prior knowledge and experimental data as constraints on the graph topology and on the dynamical properties of the models. So they come from an inference graph, we have seen at the beginning of the presentation, to delimit the interactions that can be included in the models, and also from the dynamical properties of the behaviors that have to be reproduced, and which can be reachability between observations or stability of observations, and so on. The principle of the method is that the whole set of constraints comprising the domain of admissible Boolean networks and the dynamical properties they should satisfy is represented in a single logic program expressed in Ensemble programming, such that each solution given by the solver corresponds to a distinct model, a distinct Boolean network, which is a model. So this method provides non-redundant solutions. The extension of this work is notably that we have enabled the possibility to take into account a universal property on fixed point for the synthesis. And we have also added different network perturbation settings, and we use heuristics to drive the ASP solver in different regions of the solution space. Indeed, the comprehensive set of models is often too huge to be enumerated. Thus, our aim is to sample ensembles of Boolean networks, ensembles which are ideally representative of the diversity of the whole set of models. Let's start with the first contribution, the universal property that can be now taken into account for the synthesis. Such a property not only ensures that a described behavior is in the system dynamics, as did existential constraints. A universal one ensures it's the only possible behavior. To illustrate, let's compare the meaning of existential or universal properties given the list of experimentally observed self-fact. On one hand, an existential constraint guarantees that at least one attractor of the model dynamics matches with each self-fact. On the other hand, a universal constraint ensures that every model attractor matches with at least one of the self-fact. In fact, a universal property involves by nature universal quantifiers. And Ensemble programming can address formulas implying one level of universal quantifier thanks to a technique called the saturation technique. The principles of this saturation technique and its implementation are described in our current paper. We exploit this saturation technique for answering universal constraints on the fixed points or on fixed points reachable from a given configuration of interest. Thanks to them, it's now possible to ensure that all the fixed points of the Boolean network, or those reachable from a configuration of interest, are compatible with partial observations. That means set of markers, so a set of values for some nodes of the network. And combined with mutation and previous implemented constraints, the method can leverage observations about sulfates in different mutation conditions. About the contribution on the diversity in the enumeration of the solutions, we introduce a sampling of the diversity of the models in the enumeration. It's necessary because typical solvers enumerate solutions by identifying a first solution and then by finding following ones by successive slight variations. Consequently, if we run a partial enumeration, we obtain a set of similar solutions, for example with changes on the Boolean functions of only one component. This set cannot provide a representative view of the Boolean networks that are compatible with given biological data and knowledge. It's why we introduce a strategy to sample ensembles constituted by more diverse Boolean networks. To do that, we trick heuristics of the solver Klingo to steer it towards distant solutions. Because, in fact, at each solution we randomly select a subset of viable assignments, and we ask the solver to avoid them in the next iterations. By this way, at the cost of enumeration speed, this strategy allows sampling ensembles of diverse Boolean networks. From now, the presentation will be continued by Vincent. He will present to you the simulation method and the application we did.
So hello everyone, I'm going to present you the way we simulated these ensembles of models. So to do that, we extended MABOS, which stands for Markovian Boolean Stochastic Simulator, which, as the name says, is a Boolean simulator. It's able to produce state probability trajectories. It also adds a notion of physical time to Boolean modeling, and with that is able to represent processes of different time scales, such as transcription or phosphorylation. And MABOS is also efficient, being coded in C++ and able to perform simulation in parallel. So MABOS uses a continuous time Markov process applied on a Boolean network state space. The transition rate of this Markov process is defined such that you will have a rate of activation and inactivation for every node, so for every transition. And this will allow you to represent time scales and to add this notion of time to Boolean modeling. But in this study we choose to keep this parameter to 1 so that it will stay closer to classical Boolean models and so that we'll be able to compare two standard Boolean models. So once we have this continuous time Markov process, we can simulate it and generate several stochastic trajectories, and then with these stochastic trajectories we can compute the probability distribution of each state. So the output of MABOS is are these state probability trajectories, but in this study we will mainly focus on the steady state distribution, which is just the state at the end of a simulation. And so, for example, in this case, this steady state distribution is split equally into two possible states, one where A and B are activated, and another where A and C are activated. And so to extend MABOS, what we did is allow to modify the input of MABOS and allow to give my boss an ensemble. Then every time we want to generate a trajectory, we will pick up one of the model of the ensemble, generate its trajectory, and then with all these different trajectories, we'll be able to compute the probability distribution of the ensemble. When we choose the models, we can choose to do it stochastically, but we can also sample uniformly the model space of the ensemble. Another thing that we can do also is allow to save individual results for each and every model of the ensemble. This allows to study the composition of the ensemble and to compare different models within the ensemble. And so, as an application of this pipeline, we choose to study a model published by Cohen in 2015, which represents molecular pathways regulating tumor invasion and migration. So this model has mainly two inputs: the microenvironment, which is basically the presence of TGF-beta and Notch in the medium around the cells, or the external activation of DNA damage. Then, as output, we have different cell fates. We have the activation of the epithelial mesenchymal transition. We have invasion. We have migration, which will lead to metastasis. We also have cell cycle arrest, and we have apoptosis. And so when we simulate this model with MABOS, what we get when we look at the wild type of this model is a distribution where we have around 50% of apoptosis, 25% of EMT and migration, and another 25% where cell didn't activated any of these fate nodes and so are in homeostasis. What we can look at also is a result of mutants, or in this case a double mutant, which is the activation of nodes of Notch and the inhibition of p53. In this case, this will lead to have migration 100% of the time, so all the cells will start migrating and will undergo metastasis. And this is a known experimental result that Cohen was able to reproduce. So when we built these ensembles, we thought about using the interaction graph of Cohen as a prior knowledge network. So basically as a constraint using the list of all the known interaction and their sign. Then we also added universally reachable fixed points for a specific condition. So this will allow us to see with this constraint what is the diversity of models and behaviors and phenotypes that we can simulate. We decided to generate two different ensembles, one with the reachability of Cohen's fixed points in the wild type case, so all the fixed points which are reachable in the normal condition, and another one where we take this list of fixed point reachable for the wild type, but we also add the list of fixed points under two conditions. One which is the activation of Notch, and the second one which is the inhibition of p53. And this can also help us answer the question is, does the addition of constraints from the two single mutants help us predict the phenotype produced by the double mutant? So when we simulate our ensemble, for the wild type we get a very similar result. Still some differences. We have less apoptosis and more migration, and we also have a significant amount of cells which start EMT but don't activate migration in the sequence, which was something that Cohen's model was doing. When we look at the double mutant, we still get a vast majority of cells migrating, around 2/3. But we also have this population of cells which activated EMT, but didn't go all the way to migration. Also, we have some aberrant phenotypes, like apoptosis, EMT migration. Like, this is a classic result of biology, that apoptosis and EMT migration will be mutually exclusive. When we add the two additional constraints on the single mutant, we get a very similar result for the wild type. And for the double mutation notch P53, we get less aberrant phenotype, but we get also less migration, and we get a lot more of this phenotype which activated EMT and didn't activate migration. So with these constraints only, we cannot reproduce the double mutant results. We need additional constraints. We also studied the diversity of a model by saving the individual results as I presented before. So here in this figure, every point will be one model, and its position will show the distribution of steady states. For example, one circle, a blue circle which will be on the right, will show some model which has the vast majority of its phenotype as apoptotic, and very few EMT or migration. And the opposite will be, if you look at a circle on the upper left, you will have mainly phenotypes with EMT and migration activated, but less with only EMT or apoptosis. And with the blue circle, you can see the position of the original Cohen's model inside this distribution of steady states of model. And you can see that Cohen's model is a very balanced one when we look at the wild type. But then when we look at the double mutation P53 notch, which is in orange, you can see that the orange circle of Cohen's model is at the top of this orange line. This orange line basically signifies that you have a lot less apoptosis, or most likely no apoptosis at all, and that the steady state distribution is mostly of models which are splitted between either EMT plus migration or EMT alone. And you can see that you have a diversity of distribution of balance between these two phenotypes. So, as we can see, these constraints that we used can produce very, very different models from Cohen. So again, it shows that we need more constraints if we want to build more accurate models. This also shows that the diversity we generated with our synthesis methods looks good. Looking at this plot, we can see that we don't have clusters. We don't have majority of the cells, for example, being apoptotic or something like this. We have a good distribution of behaviors on this space. So to conclude, now we can see that we can generate ensembles of models applying these new universal constraints, and that Ensemble Mabos can efficiently simulate these models. We also saw that we can visualize the diversity of our ensembles using dimensionality reduction methods such as PCA. But we need to still evaluate further the diversity of model and the quality of the heuristic we are using. So our complete pipeline is available as Jupyter notebooks on the following DOI. Bonesys, which is the library we use for synthesis of ensembles of Boolean models, is available on GitHub, as is PyMabos, the Python library we use to simulate ensembles. So with that, I thank you for your attention, and I will be happy to take questions.